Sure. Jill's presentation, I don't think we had yours yet.
Yes.
Okay, so you're good.
I'm up and running on virtual.
So you don't need my sticky.
Did you stop streaming the last one?
Yes.
Okay, and started again.
Yes.
Okay,
And
very good.
And you don't need my sticky.
Do I have to download it?
It might be running off of that.
Oh, that's fine.
Yeah,
Yeah.
just
Perfect.
run.
I just have to remember to take it.
Yep, yep.
That's my little stuff.
There we go.
You're welcome to walk around. Just make sure you have the mic so that virtual people can hear you.
Oh, right, because of that streaming.
Yep.
Okay, got it.
Yep.
Yeah, thank you for reminding me.
Yeah, no problem. And then I'm pretty sure you should be able just to push like space bar or arrow key.
Maybe not.
Yeah.
Okay.
So you should be able to just push that
Down.
button.
Yeah, okay.
And I'll read your bio and then you should be good to go.
Okay.
right, we'd like to welcome you all to this next session here. Is this making a weird noise? Let me turn it off and on. All right, let's see if that's better. All right, I get the pleasure of introducing our next speaker. So our speaker today is Jill Clapperton. Jill Clapperton, PhD, is a principal scientist and owner of Rhizoterra Inc. She is an internationally recognized educator on how to create, measure, and maintain healthy, productive soils that produce tasty, nutrient-dense food. Jill has a PhD in plant ecophysiology and worked as the Rhizoterra ecologist for the Agriculture and RE Food Canada for 16 years before founding Rhizoterra Inc. Sorry, these lines are close together. It's hard to see which one's next. Jill was a board member for the Pacific Northwest Direct Seed Association for 5 years, coordinating the Farmed Smart Grant program that certifies farms as sustainable and regenerative. Rhizoterra is a partner in our grounded growth, Harvest Today, and Prolific Earth Sciences. Jill acts as a scientific advisor to the Bionutrient Institute, supporting research for farmers that have healthy soils and grow nutrient-dense food. Rhizoterra Inc. is an international food security consulting company guiding people, organizations, and corporations as they endeavor to create and measure healthy, productive soils that grow tasty, nutrient-dense food in a way that honors our farming culture, regenerates arable land, and sustains environmental integrity. Please welcome Jill.
I gotta get this out of here. There we are. All right. I'm going to walk around because if I stand behind the podium, well, you just see the microphone and, I'm vertically challenged. So that's the issue there. Okay, if you have any questions, you can get hold of me. My email is there. And you're welcome to email me. All right, so let's see. I'm going to try, okay, it's not that one there. There we go. I like this picture. Just that you can see now the soil's habitat. So we're going to talk about habitat. So the one thing that Tony asked me to do was talk more about no-till. And I'm happy to do it. I haven't done it for a long time because there's been so much adoption of no-till that I don't think about that anymore. And I have never done any research on tillage because I never saw the value in it because we knew so much about no-till. But I was just saying at the back there, the one thing I want everybody to remember is that no-till is a system. It's not just about changing iron. You have to change the whole system. As one person I know put it when he first he went to Dwayne Beck's to learn about no-till and new ways of planting, and he said you have to sort of turn your brain inside out and upside down and then start thinking about it in a whole new way. And I think that's the thing that we have to remember here. We're talking about a system. So just because you change iron doesn't mean that you're going to change everything. You have to change the whole system. We have to think about rotation now, and we have to think about, the fact that we're creating a habitat. We have to think about roots and all these other things. So let's just preface that, because ultimately we do want to get to the point where we have a real habitat. And there's a number of reasons for that. We're going to talk about them as we go forward. So when you're standing on your ground, in a lot of cases, and some people are standing on less than 6 inches, but I'm going to talk about the organic fraction of your soil. Not the mineral soil, the organic fraction of your soil. And think about the wind. There's a good wind out there, and I know that you get a lot of wind. So what happens? Is it the heavy stuff? Is it the sand that blows away? No. It's the light fraction that blows away out of your soil. It's the organic matter that blows away. And if we looked at what was in that dust that's blowing, there'd be a lot of mites. There'd be columbo. There's all sorts of invertebrates in there because we're blowing away that organic fraction. And when we blow it away, we're blowing away our chance at soil health and about using the benefits of soil biology. So the last thing we want to do is have erosion. Plant covers stop erosion. One of the key things, let's say you've heard a lot about cover crops, so just ignore cover crops for a while. One of the best things that you can do is get a stripper header. Have tall stubble. Catch more snow, catch more rain, especially with heavy winds. You want to create a microclimate for those beautiful little plants that are starting out. Give them a microclimate. Slow that wind down. Let's try not to dry them out any more than we can. That's part of it. So that's one of the things to think about. This is side by side. This is in North Dakota. It was minus 40. I parked my car on the side of the road. It must have been minus 50 with this stinking wind chill factor. And I sort of like ran out and took these pictures and then climbed back into my car. They also stop water erosion. Having covers, any kind of cover, really stops water erosion. This is winter canola. You can imagine that I lost that part of the winter canola. This is what tillage is like. Just imagine. Look at that plow. Look, that way it looks like a moldboard plow coming into that city. Well, below ground is a whole community, a city, infrastructure. And they're spending a lot of energy to do that. A lot of energy to create the soil structure, to create these water-stable aggregates, to create this continuous soil pore network. Oh, and I am just going on autopilot here. How cool is that? And you think about this right now, you're going to destroy all that. And if I did that twice a year, plowed it under, are you going to keep rebuilding? Are you going to have the energy to keep rebuilding? Think about what has happened to us with COVID. Do we all have the energy to keep rebuilding? No, a lot of people have given up. You keep destroying things and people and organisms are just going to take it out, or either that, or they're going to die from the tillage and be destroyed, and then who rebuilds the population? So we have to think about that. When you're standing on your soil, you're standing on the rooftop of a whole other world. And as farmers, we have the opportunity to actually go ahead and manage that. The top one is sugar beets, you guessed it, with flood irrigation, leveling everything. What can you imagine? Do you think you have soil at that point? No. And you know what's happening? When do they do this? February, March. Winds blowing like crazy, soil blowing everywhere. What have we lost? Not only we're leveling, we're losing a lot of our potential for soil biological activity, our potential for soil health. The bottom one, here we are, seeding into green, planting into green. Why do you plant into green? Well, I'm from a drier climate. I started in Lethbridge, Alberta, southern Alberta. That's a 9-inch rainfall zone. Hairpinning is real. So you get a lot of dry residue. You don't have GPS tracking, or now you do, thank goodness. So that's why the stripper header is so good. I can go between the rows now easily. So we're driving between the rows, we're putting seeds in. But if I go green, that stuff cuts like butter. Now it's easy to get in there. But I have another issue, right? Then how do I deal with all the green stuff afterwards? So we're going to talk a little bit more about that. There are some really nice technological advances that have happened in the last 5 years that are going to help us grow green. And somebody mentioned about perennials and using perennials in our cropping system. That is a reality now with some of the new drone equipment, and I will show that at the end. Because I am a scientist, so I'll preface that right now. I approach things from a different viewpoint than people like Jimmy who are first a farmer. I'm first a scientist and second a farmer and a rancher. So I'm going to come at this from a slightly different viewpoint for sure. So do you have dirt or do you have soil? If you don't have things living in, a lot of things living in your soil, then you have dirt. Nobody needs dirt. We all need soil. If we're going to grow plants and we're going to grow healthy plants and great plants, we need to have soil. This is from a handful of no-till soil after 20 years. You tell me that that's not alive. That is alive. Crawling with mites. This is about decomposition. Mites eat through all the residues, create holes in it so the fungi can go in and break it down again, and so we can recycle the nutrients. We need biology to recycle nutrients. If we don't have the biology, we can't recycle the nutrients. Plants take up nutrients more effectively and efficiently when they are mediated through the biological system. So we have to create a habitat for these biology, for all the soil biota, and so that we can do that, so we can have that nutrient cycling. So that's the point of no-till. The point of no-till is that we want to create a habitat. Now that's only the first step. There are many other steps to this. I came at no-till from a very different perspective. So I come from more of a prairie background and a plant ecology background. And so when I came to the Lethbridge Research Center, I didn't even really know what no-till was. But the Lethbridge Research Center had been using the first no-till equipment, which was a Noble Blade, for 50 years at that point. And they had these really large experiments. And I can remember the first time understanding no-till. One of the scientists, he was a hydrologist, and he came in, and he had a grant, and he'd taken these humongous monoliths out of the soil. They were a meter by a meter. And they, had to excavate them out and everything. And the one in the no-till was full of pores, and there were earthworms, and then the other one was nothing. And he came to me, he goes, well, you're the rhizosphere ecologist, Jill. So do you think these earthworms and these earthworm channels have any effect on hydrology? And I was like, well, I would hope so. And he goes, well, what kind of earthworms are they? And I said, I have no idea. But let me look at that. So that started me on the whole process of looking at earthworms and really looking at more soil animals. Before that, I had really spent most of my time in plant physiology, biochemistry, looking at the quality of root exudates and looking at some of those effects. So stepped right into that. So that's the head of a pin. So you'll see that 14 of these mites will fit on the head of the pin. So that's what you were looking at in the slide before. So you saw how many there were. Lots. I'm going to remind us all that when we create healthy soil, it's really a process. So the biology arrows at the top, because they're what generate the chemical, the recycling, they also generate physical structure. Because, and I include in the biological, because plants are alive, the roots. Because roots themselves, the root exudates themselves can create or change soil structure. So for example, peas, some of you have grown peas or other legumes, they'll create a softer soil. Beans, peas, will create a much softer soil that has more tilth. Even smells, you can smell the legumes in it. You grow things like corn and the soil is much tighter, wheat, much tighter. That's actually the effect of the root exudates, driving the soil biology, which is again affecting the soil structure. So it all works together. And ultimately, that's going to create what we call soil productivity. So once again, we're creating the habitat so we have biological soil, so we can take advantage of the activities of the soil biology, which then creates soil health, and in the end, we get better productive soils. Because we have better structured soils, most of the nutrients are going to go into our food, and not so many are going to go into the environment, because that's the last thing we want. And ultimately, that's going to affect the health of our soil, of ourselves and our animals. And I will talk a lot more about that tomorrow. But right now, we're building the habitat. So let's just say for now, we have gone and we're no-till farming. Now, one of the best ways to get into this and make the transition is from a cover crop or from a pasture. And there's a reason for that. And Jimmy kind of alluded to that this morning. If I have, and some of you will have seen this, if you've tried to grow wheat after you had a really great pasture in, so you go from pasture and then the first crop you try is wheat, you will see that your wheat kind of does this. And then it might go like this, but it might just kind of stay growing really pathetically or lethargically for a long time. So think about what happens. You have now had this beautiful pasture. You've got a lot of root exudates. You've got a lot of activity, right? Because you haven't disturbed that habitat at all. And then you've gone and planted something with a puny little root system in it. Well, that is like me starving all of you for, I don't know, a couple of days, and then throwing a piece of bread in amongst you. Everybody's going to die for something to eat. Organisms are just exactly the same. That little bit of root exudate, they are all trying to get on the root. Every single one of them that are in the soil are going, let me on there. Now what happens? Well, if we're fighting over a piece of bread in the middle, what happens? People start slugging each other, dragging each other off, and the whole thing turns into a melee. Well, microorganisms are not that much different, except they poison each other. So then they start gassing, producing hydrogen cyanide gas, and various other things. So it's like, you're out of the way, I'm taking over. So how do you think the plant root responds to that? The same way you or I would if we were getting gassed. Plant root's like, whoa, slow down. And some of those roots are dying, and then the puny little root systems are dying back even more, and the whole thing goes into this downward spiral. So, once again, I said it's not just about the iron, it's about the system. We start from a beautiful, stable environment, and we start into no-till. The last thing you plant is a puny little root system. You need to plant something better. You are in an area where you can grow warm season crops. The first thing you do is grow a warm season crop. Why? Because warm seasons really want a lot of biology on their root system, and so they actually are trying to attract it in the first place. So you grow a warm season crop, and then you work your system from there. So the first thing, and if you're not starting from transitioning from that, maybe you're transitioning from a cover crop, please, still don't go into wheat after a cover crop. Go into a legume or go into a warm season or go into a broadleaf. It will go a lot better for you. Because these livestock need a food source. And that's what we're trying to do. We're trying to create a habitat so that they can create this environment, but we still have to feed them. So healthy soil is a habitat, and I wanted you to see, just have a look at that middle image. That middle image, does that not look like a road map? Can you see all the channels and the tunnels and things like that? That is what soil structure should look like. We used to do that. One of the best ways that image there comes from doing a CAT scan. And so at the University of Saskatchewan, one of the first CAT scans they got at the hospital was in conjunction with the University of Saskatchewan. And so what used to happen is the grad students, on off times to the hospital, would run down and put soil cores in the CAT scan. And then if there was an emergency, everybody would be running off trying to, wipe up the soil and clean everything up so that a patient could come in. But that's how some of these images are created with CAT scan. Now, this one here, that's all columbolum poop. So that's an invertebrate, and that is just actually poop. And in the top sort of 3 inches of your soil, most of that will be all biology, all of it. And the biology are creating it, soil structure. So as they're eating up those residues and pooping out the concentrated nutrients, they're creating more soil structure too. And they're creating it right at the soil surface. And then if you look at this last one over here, you can see that there's lots of room. When we, it's like you have to do the Honey, I Shrunk the Kids thing. Because everybody's thinking, well, that's just a tiny space. Well, if you're already really tiny, that's actually a really big space. And you can move around in there pretty freely. And that's what we want, because a lot of the things that are predators, and the bacteria and the fungi, none of them can really burrow. So they have to move around the soil particles. So a lot of times they create more soil particles. They create bigger ones and bigger ones and bigger ones. And the nematodes and the microarthropods and the protozoa that eat a lot of the bacteria, and the nematodes that eat a lot of the fungi, they can't work if they don't have good soil structure, because they can't burrow at all. So what can happen, and this happened in a no-till soil in near Genesee, Idaho. So what happened was, all of a sudden, farmer says to me, he goes, Jill, you kind of come and look at this, like there's something really wrong. It's like my plants are always starving. And I am just pouring the fertilizer on them all the time. What is going on? Well, I do biology. I start looking at the biology and I go, this is some of the most biologically active soil I've ever seen from a bacterial and a fungi standpoint, but there's no predators. I don't see any protozoa. I don't get any nematode, nothing. So what was happening? Your bacteria and your fungi can grow whole new generations between 18 hours and 48 hours. And they can use up all the nutrients in there. And so the bacteria and the fungi were growing so great, long-term no-till soil, great habitat, but not a habitat for protozoa. Why? Because there was still this compaction layer because of the wet springs they'd been having going in, sidewall compaction from the disc drill, trying to get the crop in early all the time. We had to rethink the whole system at that point. And that was when had to go and borrow somebody's tillage implement. And so we did two things. We did the tillage. Now, at the fuel prices that I see here, at using up, pretty much 13, 14 gallons an acre, that was a very expensive little experiment. And then we then we did seed, and so then we said, okay, let's do a cover crop, see if we can break it up, and there hadn't been cows on that farm for almost 30 years. On come the cows. Cover crops, cows, everything else. We solved it with the tillage, and a year later we solved it with the cover crops. Which one was more economical? The cover crops were by far more economical. So, I just want you to understand again, it's about the system. We start creating the system, but you can't just not do, yes?
On that last slide,
Mm-hmm.
is that males have a scan of healthy soil?
Yeah, it is, yeah.
So, the thing that I comment on the irrigation system and other things, so I some of those, or is that something to worry about in that scenario?
You can have reactions with soils. Yeah, I mean, sometimes with things that are in the water, we can have reactions with things that are already in our soils, for sure. Did it plug it up? Probably not. But again, if I have a good selection of ecosystem services. So in other words, if I have a diversity of organisms, then if I start plugging things up, some other things will step in and start breaking those things down. I think there was a question this morning, I think it was actually your question, about high pH soils. Some of the things we can do with things like that to keep that whole system going is actually by using plants that actually exude acids. So things like lupins and things like buckwheat and some other things that actually use up the calcium. Some forage rapes, for example, will use up the calcium, will suck it up. Probably buckwheat is the best one at that. and then at the same time, they're leaking a lot of acids into the system. So once again, so we can start breaking up some of those reactions. But plants will also do that themselves, like not just special plants, but some of the plants that we grow. And that's the important of the root exudates, is the root exudates themselves, once again, will change some of that. Because if they're not getting, let's say they can't push through and they can't create that soil structure, they're going to leak out acids as well. They're going to leak out anions, they're going to leak out cations, but they're going to leak things out that allow them to get the nutrients that they need. One of the ways that you can measure this, people go, okay, how do I know if my soils are biologically active? This is a fun way, and a way that you can also learn. It's actually a real test, though. So this is what people didn't understand, is a long time ago, there was actually a test where you would buy strips, cotton strips that were milled at a specific mill in England. And then they had a tensiometer on the end, so in other words, it just stretched it, and then measured the amount of tension before it broke. And that would actually give you an idea of your rate of decomposition. So this is called the cotton strip test. This is based on the cotton strip test, but this is about, well, how much of my undies are left in the end. And so it's not, obviously it's not quantitative in any way. It's just like, oh, this one didn't do anything, this one did something. You leave them both in for the same amount of time, dig them up and go, okay, I have decomposition. Why is that? Because fungi break down cotton. Because cotton is a lignin fiber. And so if you want to know how active the fungal community is and the arthropods in your community are, then you bury underwear and cotton underwear, and away you go. Roots will move down old earthworm channels, but it will also allow your soils to mix. So this is a gray-wooded soil, and you can see that there's a root coming down there. There's another reason. So now you've got this mixing. And our objective in retaining that habitat is now that we're not disturbing the soil, it's about creating this habitat, like this really deep rooting zone. Now we're going for mixing, because now we're having more biological activity. We'll see more earthworms, we'll see more other things, and so now we can take advantage of this mixing activity, so we create better soils. Now, this is an experiment that I did, and all I want you to see there is rotation 3 and 5 were organic, 1, 3, and 5 were organic rotations. They all had tillage involved. Earthworms, no matter where you go in the world, are very, very sensitive to tillage. That includes, you can go to Germany, same thing, where you have tillage, which is the direct seeding there, or pasture, and pasture is more of a diversity effect. You will always, always have more earthworms. Earthworms are one of your best indicator of how well you're doing at creating a habitat. Because at the end of your soil food web are the earthworms. They're the top of the chain. And so if you get everything right beforehand, you're going to get earthworms. This is an experiment we did at Lethbridge, and you can see the same thing. Now, the interesting thing was that in autumn tillage, we learned that fall tillage was the most detrimental of anything that we could do. And at first we thought, well, it shouldn't be that bad because earthworms actually rest. But the problem is that we're not tilling when they're resting. If you get good snow cover and your soils are still a bit moist and whatnot, the earthworms are still active. And if they're resting, then all we load themselves with antifreeze. And that's how they load themselves with antifreeze, by making this wall. But then when we rumble across it, we crack it. And then they become subject to water and temperature, and they die. And so we killed off most of them, and they didn't recover. But in the spring, they actually recover. This is why we want earthworms. Look at how we've got that mixing. Darwin actually called earthworms nature's plow. So let's look at this one here. Now you can see that first, we're looking here, you can see the one on the left, that's bad structure. I don't have really good structure there. And that plant is working really hard to go through it. And when that plant uses a lot of energy to go through that soil, it's wasting energy on trying to get nutrients with its roots. So it doesn't have enough energy to grow a lot more shoots. It is just trying to push through the soil. So it's creating a little bit of soil structure, but mostly it's spending a lot of time getting nutrients. And you can see that it hasn't grown any lateral roots like the other one, right? Plants don't grow lateral roots. They're using way too much energy because they only grow lateral roots when they're not using a whole lot of energy. When the resources are really good, they grow lateral roots. Then they create more soil structure, then they get more nutrients, then we get more above ground. So that gives you an example of the soils. Now, I'm not too worried about that platy one because you can still see that root's going through, but that root is using way too much energy to go through that plate. So how do we build more of a habitat? How do we do that? Now, I know we probably got some perhaps some vegetable growers in here. One of those things with that is that if you're growing potatoes, I work with potato and onion growers a lot in the Columbia Basin area of Washington State, and we do tillage. I mean, we have to lift the onions, have to lift the potatoes. We're going to do tillage. Sugar beets are exactly the same. We're going to do some tillage. But I can tell you on the onion field, we've got halfway through the onion field, and the drill comes in and starts seeding the cover crop. Because that's a way to mitigate some of that compaction. And we've got 20 tons of onions coming off in each one of the trucks. And so those trucks are getting loaded to 20 tons before they take off of the field. And so we have a lot of compaction, and the only way to overcome it is by coming in right away with a cover crop. In this case, we are interseeding here. It's all about the roots. And you can see what's happened there. That's a corn plant. You can see which one has a better root structure, the one that's growing in better structured soil, the one that is not having sedimentation. And yeah, you can build your own little rainfall simulator out of drink bottles. And pastures, because I know there are some grazers, I met some last night, are not immune. They're not immune to erosion. You don't manage your grazing well enough, you're going to have erosion. We can't have water erosion either. We want everything going through. You want to capture all the water, and especially in dry environments, we cannot afford to lose a drop. So what are we going to do? We're going to grow more roots. So you change iron, you start making a habitat. Well, now you have to really build that habitat, and the only way to do that is with roots. Now, we've talked a lot about cover crops in the last little while. I come from really dry areas. In Washington State, we have winter rainfall. So we're planting our crops in March and April, and sometimes at the end of February. And we will get a little bit of rainfall after that. We might get the odd shower in June, and then we dry out. And we won't get anything more until probably about October. And then we're cold in November. So I have a short window. We tried a lot of cover crops because obviously I'm an advocate for that. I started cover cropping because I hate fallow. I think fallow is a waste of time. Fallow is an opportunity to starve out your soil biology. It's not a rest, it's a starvation. And so how do we get rid of that? Well, we grow a cover crop or we graze in between. That was the way, so I started that. And for that system, it works great. But for dry systems, I'm going to tell you that I think companions are is something that I can do. I think companions are something that is easier to do. I think it works in the dry climates. I think it works in cold. Now, you're a little different because you're not as cold, but you get farther north, we have more cold. And I am not going to grow in, I'm going to have something dormant over winter, but I have a very short window to get a cover crop in the fall. A very short window. And then that thing is only going to be about this big. Well, that's okay if that's what I can do, but with companions, I can actually harvest my crop, have some companions in there, or if I can get in with the drill and get some moisture, I'll have something a bit bigger that might take me through the winter, might hold my soil a lot better. So I'm going to talk more about companions. And you can see when you get companions, you actually have more roots generally. I'm also going to build more carbon. I'm going to build more organic matter. I'm going to build my water holding capacity and my structure faster with companions than I am necessarily with a cover crop. And the most important thing is, it's not about the residue you have on the surface. It's not about how much stuff I grew above ground. It's how much stuff did I grow below ground. Because how are you feeding all the structure? How am I creating all the structure? I know I can't just create it with the drill. So how do I create it? I create it with root exudates. I create it with roots. I create it with root carbon. And the most available source of carbon is from the roots. That's what's going to build your soil. The most active, biologically active part of your crop is the root zone. It's not between the roots, it's in that root. So if I'm growing companions, I'm filling up that whole field with roots, I have the most biologically active part of my soil. I have photosynthesis, I am feeding the below ground. Photosynthesis is about feeding the below ground, not just about the above ground, it's about the below ground too. I can't shoot minerals up if I am not pulling stuff out of the ground. So I need both. This is a case of sunflowers. This is in southern Alberta, against the Rocky Mountains, in Claresholm, Alberta. We have faba beans and crimson clover between the rows of the sunflowers. We still took a beautiful sunflower crop for spits. And at the same time, we filled the whole profile with a lot more carbon, and we were building carbon, we were building water retention, we were building water, and we were building infiltration. So one of the easiest measures of whether you've got good stable aggregates and you're building a beautiful healthy soil is your rate of infiltration. Because how fast the water moves in is directly proportional to the structure. I also oversow pastures. We call it stitching. And as soon as I start adding broadleaves into the pasture, not only do the cattle improve, but we also start building more water retention, more water holding capacity, better infiltration, and we start having the system work. There's in ecology, it's called disruptive hypothesis. So what it is, it's about when you have a stable ecosystem, how do you sort of slap it in to productivity? Well, you can light a match, or you can put some other species in there that they actually have to compete against, and all of a sudden it's like, oh, I actually have to work now. I can't just rest on my laurels. I have to get going. And that will also really improve your soil structure. And in fact, we can, within 18 months, we can have a completely different soil structure in pastures by just seeding a diversity of broadleaves. And I'll show a picture of that tomorrow. And the most important thing is having legumes, because legumes are what really push the carbon down. They're what stimulate more photosynthesis. Yeah, are you? Sure. Direct seeding, and you need a disc drill, by the way. And so there is a way to do this. So if you're going to direct seed a pasture, you need to graze it down first. You might run a bit of a harrow over to get anything that's not rooted very well out of there before you take your drill in. The next thing is you can do two things. You can wait till your grasses senesce a little bit. If you get sort of a bit of rain in June, you can actually go in and seed warm seasons like millets and even some cowpeas, but I wouldn't seed cowpeas. I would probably seed beans like mung beans. And I might even seed lentils and chickpeas. In my case, probably chickpeas, garbs. And I put them in, and animals love garbanzo beans, by the way. If you grow garbs, you know that deer absolutely can't keep their little teeth off them. But I grow the desies, so I'm going to grow more of a wild type chickpea. I'm going to grow lentils, some of the cheaper lentils in there. And you'd be surprised, because with that heat, you'll get something, and then you'll get the millets, and then you get some warm seasons. It's one of the easiest ways to get warm seasons into your system. Proso millet is probably the coldest tolerant millet that you can get. I also really like lupins, sweet lupins, especially if you have your calcareous soils. Lupins are really good at changing that a little bit. They're a bit harder to establish, but you can do it. So we talked about that. So root channels. So you can see, again, that's the carbon from the roots. So a lot of people say, well, I can't see the carbon from the roots. That is the oxidized carbon from the roots in that soil. That's what they leaked over the growing season. So you imagine I put more roots in there, how I'm going to change the colors of the soils, right? I'm going to start to see color change. And then there's the mycorrhizal network. If we wash all the soil out and we have a good healthy soil, that's what the mycorrhizal network looks like when it's magnified over 1,000 times. That's all carbon. That's all structure. So what's going on there? This is the point you all had a little bit of a chance. Everybody's starting to wane now a little bit for the afternoon. What's happening there? Why do we have those brown spots? And I won't say anything more until somebody ventures a guess, so. Pardon me? Yeah, that's part of it. What else? Yeah, exactly. So what's the underlying cause of the erosion? What do I not have in there that is causing me to not be able to grow anything there? That's part of it. I love it. What else? Biology, yeah. I don't have biology because I don't have any carbon. I have no water holding capacity on eroded knolls. You don't get a sense of how steep that is, but that's actually really steep. It's in the Palouse country in Washington. And those are all eroded knolls. And I'm not growing anything on any of them because they've eroded. They've washed, they've blown away, and all that organic carbon is gone. So we started, now where is that slide? Okay. So we started adding organic matter in pellets. So I worked with a composter to create pellets. And when you create these pellets, this is what is essentially what's happening in your organic matter. When you create the pellets from compost, these compost pellets had an 87% water holding capacity. So it was like putting a little sponge in with the seed, so that when I got rain, it would hold. And I wanted to put it in furrow. I didn't want to just broadcast it on. I wanted to get it in the ground with the seed because I wanted that sponge, right? I wanted something to hold on to it. Plus I get the nutrients, plus I get the biology, all of those things that people talked about, plus I increase the cation exchange capacity. So I'm really putting this seed into a beautiful product. Well, when we did that, that's 40 pounds to the acre. And all of a sudden we didn't have that anymore. It's a slightly different angle. You can still see that there were some issues there, but mostly it looked really good.
Pelletizing with compost,
Yeah.
what changed in the mention? Did you put some unit in it, or was there some?
No, it actually was just straight compost put through a pellet mill and was combined with oil, a little bit of canola oil, raw canola oil, because that was the cheapest we could get.
That versus just straight compost, apparently something changed dramatically.
Well, part of it is when you just use compost that isn't pelleted and you try putting that in furrow, first of all, it clogs your drill really bad. It can. You get dusted up pretty badly, like you pretty much just end up wearing half the compost that day when you do it. And you don't get enough in. It's really hard to put 40 pounds to the acre of soft, fluffy compost into the furrow. But with the pellet, I concentrate now. I've pelleted that compost, so it's all compacted. Now it flows through my drill really easily. I'm flowing it with my fertilizer, and I'm putting the seed above it.
I missed that part that you actually furrowing it.
I was in furrowing it through my drill. And that's why no, you can't. If we didn't pellet it, no, you'd never do that. And there are some people in Colorado that are doing that with manure as well. They're pelleting manure. Again, a really nice product. If you have a problem with eroded soils or you're trying to build organic matter really fast, this is a really nice way to do it. Now, increasingly in the Pacific Northwest with climate change, we are experiencing much hotter Junes, 100-degree Junes. Now, that's nothing new to people here, but 100 degrees in June is really news to us. And the first year it happened, I had everybody's growing cool season plants, right? We grow cool seasons. So you get 100 degrees in June, and your wheat really tanks it. And everybody's going, oh, it's a drought. And I'm like, I don't know about you, but I dug up my plants, and I still had moisture. I had dead plants with moisture. How does that happen? You want to cry when that happens. I'll be honest, I really wanted to cry. Because I dug them up and it was still moist, the soil was still moist, and the wheat was gone. And I was going to, I was like, okay, now what do I do? Well, wait a minute. I have moisture. It is now warm season time. So what did I do? I planted sunflowers. And I took a sunflower crop. So this is monoculture turnips. This is monoculture radish. That's a mix. What happened? Same day, same place. Why the mix so green on the same day? Yeah, it's diversity. Yeah. It's also about the mix of the roots. When the roots mix like that, they share. And that's what people don't understand. They like a community. And communities share. They're sharing water, they're sharing nutrients, they're sharing. And you put your hand under there and it's moist and cool. Here's what wheat stubble, that's the drawdown on wheat stubble. And you can see the timeline. Same timeline on a mix. You don't get the drawdown. That whole idea that I can't grow companions because they're going to rob my crop of moisture, that is true when I start. Absolutely. When my soils don't have the structure, when I don't have the biology, when I don't have the moisture holding capacity, that is absolutely true. But when I do, this happens. And I don't use as much water, and it's not as hot. Now, there's another reason for that. When the soil gets hot, it means roots get hot. When roots get hot, you have no more transpiration. They can't move a thing. So now I think about this with my orchard. With my orchard, I started mulching my orchard. My neighbor on Apple Fest had this great maze made with all these straw bales, and I just said, hey, can I buy your straw bales? And he goes, well, I don't know what you can do with them. Here, have them. Great, I said. I mulched my whole orchard. Mulched it. Created these little collars that I put around the trees, and I mulched it 6 inches high. I mulched every tree that high. Never, ever had a problem with my Honeycrisp with bitter pit. Because all of a sudden, roots never got hot. Calcium and boron was just pumping up there no matter how hot it got outside. I maintained the transpiration stream. That's why you mulch. That's why you keep the ground covered. That's why you want cooler temperatures. Because you want to maintain moisture. Same day, cover crop, 87.6. You're over 100 degrees in the monoculture. Which one is frying? That's still pretty hot. We're going to hold that armor on the soil. Yes, it makes it a bit cooler in the spring. No questions asked at first. But once your system gets going, it actually makes it hotter. Because the biology give off energy in the spring. Because you know what? They are active. They're active at 35 degrees. They're starting to come out. Earthworms are starting to get active. By 40 degrees, they're in full mode. Your soil's at 40 degrees, your soils are happening. Because cold, moist soils carry more oxygen. Cold water carries more oxygen. They are busy. We maintain that temperature a little bit, they get busy. That means the soils heat up faster. That means we get to use the idea of no-till. And you can see here, as long as we stay into the 70 degrees and sort of up to the 80 degrees, we can maintain biological function. But once we start to get over that, we are toasting things. Temperature affects rooting. And I forgot to convert this. I apologize. 20 degrees is 68 degrees Fahrenheit, just so you know. So 20 degrees Celsius, 68 degrees Fahrenheit is about perfect for rooting, especially for warm seasons. 15 degrees works just fine as well, and that's around, your 55 degrees mark. And then we get to 10 degrees, and things are getting a little tricky. We go a little hotter, and we have some of the same issues. So you want to actually stay in the proper zone. And that's why planting to temperature, if we don't have too many acres, is really important. Now, this is the part I told you I'd come to. This is alfalfa, and you can see that there's something else in there, right? Because you can see the rows in there. That's actually clover bit. Okay. Now we have the row mow. Oh, look, there's corn. This is from Dawn Equipment. And so the row mow is from Dawn Equipment, as is the row mulcher. And so these, if you have to go into a wider row spacing, but it actually will mow between your rows. I love this. This is the in-row roller. There we go. There we go again. It's a beautiful piece of equipment that allows us to create. We can roll things down, we can roll the residue down, we can roll things down, but we can also mow them. I'm more of a mower because I don't get, the residue I grow doesn't get brittle enough to roll very well. I'm a terrible roller crimper. I mean, I roll it and then it just sprouts back up and then I end up spraying it out or something. So I must admit that I tend to go to mowing. So 5 years of no-till. This is a farm near Reardon, Washington. I had 80 acres there. 60 units of Solution 32. One of your biggest problems, and I'm going to talk about this in the next 2 days, anhydrous ammonia in dry climates, even in wet climates. Bad deal. I know it's cheaper, and I know how expensive it is this year. I blew all my old stuff, all my stockpile of nitrogen on my winter wheat. And I'm in the drought, so who knows, I might just have blown it all. I'm not sure. And buying more is going to be super expensive. 15 pounds of Tiger 90. That's elemental sulfur in furrow. Why am I doing that? Wheat and sulfur. Most of our soils are sulfur deficient. I use elemental sulfur because it's the only thing that builds up sulfur oxidizing microorganisms. If I use thiazole or any kind of soluble sulfur, it will not build my microbes. It will